Josh, we talk about antibody drug conjugates, but it feels to me that generally we talk about antibody and drug. And I often feel that the poor linker bit in the middle is almost like the unloved part of the ADC package. Is that a fair assessment?
It is and it isn't. I think linker technology at the moment is probably the most developed after the antibody itself. Conjugation probably a close third. We're seeing people really understand what linker technology is. My main caveat is that it's in isolation. We are thinking about the antibody. We're thinking about the conjugation position. We're thinking about the linker, and then we're thinking about the payload, when in fact, we need to be thinking about it all. We know that the biophysical properties that linker brings to the antibody has a massive effect. If it's hydrophobic, we're going to have a shorter half-life and we're going to have other biophysical issues. If it's hydrophilic, okay, that's great. That's what we want to do, but we need to understand exactly what is going on with that characteristic to understand the whole entity itself. I mean, then you've got to go onto the side of things like cleavable versus non-cleavable. Don't know how you feel about that and where you sit on it.
Well, I'll be honest, it's not an area that I'm really hot on. And I was going to ask you exactly the same question, because intrinsically you can't help but think, well, surely a cleavable link is better because you need to liberate your drug when you get it into a cell. So, I mean, take me through your thinking on that. Cleavable versus non-cleavable. Where'd you go?
As a just general ballpark rule, I would say cleavable is better as long as you're cleaving on the right target. If you end up cleaving everywhere, then you're not selected anymore. So you probably want to move away from that. Non-cleavable does work. I think it's very much target specific. If we see myeloid, then maybe non-cleavable will be better. We don't want any bystander effect or any off-target cleavage where we end up just with a payload floating around in the peripheral blood. If we're on a surface target, like a metabolic target, like a GLP-1, then we probably want to stay away from a cleavable as well. We want to keep that entity together. But it's very much target specific and disease specific. We need to think about everything that's going on, and it's not one shoe fits all, or we're looking for one entity to the other.
So I guess just for those who aren't used to working in the ADC space, intrinsically a non-cleavable linker on an ADC seems a bit counterproductive because, as I say, you're not going to liberate your payload. But that's not really the case, is it? Because you still effectively, whilst it's classed as a non-cleavable, the drug still gets cleaved. I mean, do you want to comment on that?
You will cleave. You will cleave the linker. Well, actually, do you know what? To be truth, you probably won't. You won't cleave the linker. You will degrade around everything else. So you go through, once the antibody goes into the lysosome and goes through proteolytic degradation, that's when you release your payload. And it's the linker payload itself. And normally you have a maleimide conjugation in this situation. So you'll have the cysteine attached to the maleimide, attached to the linker, and attached to the payload, which is a non-permeable as well. So yeah, you will cleave it, but it will be the whole antibody that degrades beforehand.
But it's then understanding that, isn't it? Because you then need to build that into your design strategy for your payload to make sure that it still has intrinsic potency, even with that remnant, however much is left, that remnant of that tag still attached.
Yeah, absolutely. And I think that comes on to a really nice point in terms of the conjugation position itself and how we build the conjugation in. We're seeing a lot of development within the conjugation side of things. We know how much this affects the biophysical properties. If you take trastuzumab versus Kadcyla, we're looking at a half-life drop from 30 days to 5 days, and we need to really understand exactly what that is. And we're seeing a lot of technology come through, whether it's things like GlycoConnect or whether it's non-canonical amino acid incorporation, or even thiamab-based technologies where you're using an engineered cysteine. We know that the homogeneity of the antibody drug conjugate is really paramount for these properties.
And I'm really interested in this because I think that idea of bringing in non-canonical amino acids, non-natural amino acids, bioconjugation, site-specific conjugation, that just feels a more rational approach to controlling things like heterogeneity of your ADC, drug-antibody ratio. So tightly controlling that DAR, but being able to put on a specific number of not just maleimide through a cysteine linker, but through actually being able to proactively engineer how, where, and how many times your link is going to conjugate with your antibody.
Yeah, absolutely. I mean, you're from a small molecule world, Alan, so you understand that if you try to get something through the clinic, which is 25% of what you say it is, and then the 75% is something that's similar but not the same, the FDA or any of the governing bodies are going to look at that and go, hang on, you can't just decide that this is going to go through the clinic. But that's sort of where we are at the moment with average DARs and interchain disulfide conjugations or NHS-mediated conjugations. We really need to dial in that not only from a manufacturing position, but from a predictability position. If we understand exactly what that one molecule does, then we can understand what the whole population of that drug does, as opposed to the DAR2 or the DAR4 fragment or the DAR6 fraction within that sample. It's a really key understanding that we need that is limited at the moment. So
I think it is, because patient response to these agents is heterogeneous anyway, just because of intra-patient variability. And then when you start layering on, well, we've got a heterogeneous drug in a heterogeneous patient population, how on earth are our clinician colleagues going to understand how much to dose, to whom, when, to get that therapeutic benefit? I think the more we can control that, surely that's got to make the overall therapeutic efficiency of these molecules better.
Yeah, absolutely. I think that this is the way that we're going to start to see it evolve. And the other thing is, if we all dial in it back to this biophysical properties, we really need to understand the biophysical implications of the payload and the linker as a single entity, and not just we're using a val-cit linker, so we know that this is what the biophysical properties are, even though the payload might be different. And it's that interplay between all of the components, the conjugation, the linker, and the payload, that we really need to understand. So to bring it right the way back, linkers are definitely important, but we need to think of it as a whole package as opposed to just the linker by itself.
But I think also thinking about where that cleavage occurs is important. I was listening to an interesting argument between two people working in the field about whether you want intracellular cleavage or extracellular cleavage. Where do you want your linker to be cleaved? I mean, I guess there are pros and cons of both, but understanding then the medicinal chemistry of your payload. Do you have high permeability? If it's cleaved extracellularly, you can perhaps link into different extracellular matrix processes to liberate your payload that may be disease-specific, be it tumor-specific or otherwise. But then you've got to have the right medicinal chemistry in your payload to make sure that you have high permeability, low efflux. I mean, what are your thoughts on intra versus extracellular cleavage?
I think it depends on the mechanism of action of the target itself. So I think extracellular cleavage can be brilliant, particularly if you're targeting a membrane-bound protein, something like a GPCR. We know GPCRs are still the largest drug class out there at the moment. We know what that has. So if we want to target something, it opens up a new door to us. At the moment, if we're going via a cytotoxic payload, intracellular for me is better. I wouldn't want something that is non-selective and highly cytotoxic released externally. Even if it is in the tumor microenvironment, the more we can contain that release, the better it will be. But I think it's very much mechanism dependent. Yeah. And I think one of the nice things we're seeing at the moment is the real thought process around new mechanisms of action, not just for the payload, but for the linker itself. There is another mechanism of action that we're introducing into a molecule that already has the antibody mechanism of action and the payload mechanism of action. There's a third entity in there now, and really knowing how that interplay is really key.
And I guess we've not touched on completely non-cleavable linkers, so they're really stable linkers. So as you say, on an extracellular target, you can bind to your antibody target and then agonize or antagonize a membrane surface receptor and keep those two completely fixed. And I think that's another area that we're not really exploring at the moment.
No, not really. We're starting to see a bit coming through, but not to the same extent as what we'd expect. If you can immobilize your antibody onto the surface of your target cell and then basically have a non-cleavable linker that is able to attach into a GPCR or an ion channel, then actually we've created this targeting mechanism that works almost like a crane going into a target, doesn't it? So it's an anchor point, and it means that we're not ended up with that small molecule floating around the peripheral blood. We've got it targeted to the target itself.
So linkers are no longer the unloved child of the ADC world?
No, I don't think so. I think we're going to see an evolution of them quite quickly.
It's going to be interesting to see that evolution, isn't it?
Absolutely.